Hello, and I am Mrs. Pickman in case you have forgotten. But today we're going to talk about the DNA molecules. So, with the notes, you have learned that DNA has three components. And I want you to think in your head, what are those three components? DNA, which is deoxyribose nucleic acid, is made up of a nucleotide. Now, nucleotides have three parts. The first part is the sugar, or the deoxyribose, and I'm going to represent that with this white bead right here. The second part of a nucleotide is called a phosphate, which the sugar and the phosphate make the backbone, or the structure of the DNA. The phosphate is this red bead right here. Now, each nucleotide has to have a nitrogen base, and there are 4 nitrogen bases total: adenine, thymine, cytosine, and guanine. Now remember that each base has to have a complementary pair. So adenine has to go with thymine. Cytosine has to go with guanine. They cannot switch. So let's build a nucleotide. Nucleotides, remember, have 3 parts, so I should have 3 beads. I have my sugar and my phosphate, which make the backbone, and then I have my nitrogen base. So this is a nucleotide, which is also be called a monomer, which mono means one. So this is a monomer, or a single nucleotide. Now, DNA cannot just be one nucleotide. It's made up of millions of nucleotides. So let's make another one. We're going to make the complementary nucleotide, which is adenine goes with thymine. So these two are now complementary nucleotides. But if you look, they cannot match up together. What holds these nucleotides together are going to be something called a hydrogen bond. Now, the hydrogen bond we're going to represent with this clear little bar. So, if I add the hydrogen bond to my nucleotides, now I have a chunk of DNA. So let's keep this going. I'm going to build another nucleotide, because remember, all of these nucleotides are what makes the DNA. So in order to have a nucleotide, I have to have a sugar, a phosphate, and then a nitrogen base. Now, this time, I'm going to use cytosine. And I'm also going to make another one, because I have to make the complementary strand. So I have to make guanine. Now, they still don't match. I'm missing a component, and that component is a hydrogen bond. Remember, this is the hydrogen bond. So if I match these together, I now have another chunk of DNA. And I can combine these together. But the sugar, phosphate, and then I have to have another sugar. So I should be making a chain. Now, as you can see, I'm starting to make DNA. So I'm going to keep going, and I'll be right back. If you see, I've made a bigger strand of DNA. Now, if I start to twist my DNA, you can see that it's starting to make that famous shape, which is known as a double helix. Now, there's something I want you to notice. If you look on this side, there's a blue one on this side, but that's not the same down here. And that's okay. It does not matter which side the adenine and thymine is on, or the cytosine and the guanine. As long as they are matching together, that's all that matters. So that's DNA.